To really get osmosis, you need this one concept, tonicity. It sounds complicated, but it's really just a way to compare the concentration of a solution to the concentration inside a cell. It tells you whether that solution will make a cell gain or lose water. You've got three scenarios: isotonic, where it's balanced; hypertonic, where the solution is more concentrated; and hypotonic, where it's less concentrated. In this slide just shows you the consequences perfectly. I mean, look at that animal cell. You put it in a hypertonic, salty solution, water rushes out and it shrivels up. But put it in a hypotonic solution, like pure water, and water rushes in so fast that it literally bursts. Now look at the plant cell. Its tough cell wall completely changes the game. In that same hypotonic solution that made the animal cell explode, the plant cell just gets nice and firm, turgid. And that's exactly what a plant needs to stand up straight. It's just a perfect example of how structure dictates function.
So, we've gone way down into the microscopic mechanics. But let's zoom back out. Why does any of this matter in the grand scheme of things? How does this connect to our world?
Well, for one, those proteins we saw in the mosaic, they're not just doors and tunnels. They are the ultimate multitaskers. They can be enzymes, they can receive signals from the outside world, they act like little ID tags so cells can recognize each other. They do so much. They're really the workhorses of the membrane.
And understanding these proteins isn't just academic, it's medically critical. Take HIV, for instance. To infect one of our cells, the virus absolutely has to latch on to a specific type of protein on the cell surface. There are actually a small number of people who are naturally immune because their cells are missing that exact protein, so the virus just can't get in. That knowledge is the entire basis for developing drugs that block that exact same interaction.
Okay, let's wrap this all up. What have we learned? The cell membrane isn't just a baggie holding the cell's guts in. It is a fluid, active, intelligent barrier, a fluid mosaic. It uses both passive, no-energy transport, and active, energy-burning transport to control every single thing that comes and goes. And in the end, that power to choose, that selective permeability, is the very thing that draws the line between the random world outside and the incredible, organized miracle of life inside.
So I'll leave you with this final thought to chew on. This fluid, constantly shifting barrier is what defines every single living cell on the planet. So what does it really mean, both biologically and maybe even philosophically, that life itself chooses to exist right on such a dynamic, ever-changing, and permeable edge? Pretty amazing stuff. Thanks for watching.